In the similar manner, I am not discussing with L5 and L10. L5 where IC50 value is 125 and 10 150 only. So the extract is very important. Amount is also very important in lung cancer activity. And this is the first time with this cell line, this has been performed. Now my next is the diabetic activity that is done with only 1 ml. Here is, I am not showing the with variation what and how the anti-diabetic activity is changing, but this is the in vivo study, also in vitro study, and percentage of extract is again 25%. Flower of Plumeria obtusa as well as Tabernae montana both are taken, and similar manner we have synthesized the same concentration whatever we have taken with Azeritica indica, the metal precursor we have taken same. Only the extract is changing, where is flower extract, and these are the characterization how band gap and lambda max. So lambda max is not varying and not too much variation in the band gap, but the anti-diabetic property is changed due to presence of specific phytochemical present in the flower extract. These are the, I am escaping other characterization part. These are the morphology. This is with Tabernae montana and this is Plumeria of the missile. We have get different morphology since the phytochemical are different in both flowers, so the capping efficiency or we say stabilizing efficiency both are the different, so we getting the different morphology. The in vivo study has been done on C57BL6 male mice, weight is 25 and the age of the mice is 8 to 12 week, and these are the temperature in which these animals are kept. This study is done with collaboration with INMAS, DRDO, Delhi. And we have done also in vitro study. In vitro study has been done with respect to percentage inhibition of alpha amylase activity with both synthesized particle. This study is not done with comparative with only extract. This study is only done with only synthesized zinc oxide with extract, not with chemical method, not with only extract. And we have compare our result with the standard anti-diabetic drug, that is a carbose. Since the anti-diabetic activity of with Tabernae montana is lesser than the Plumeria optimisa, but the standard drug which is used in this treatment of diabetic, and all these studies are regarding diabetic 2. As we know, there are two types of the diabetic disease, type 1 and type 2. These are oriented study with type 2. And this is the sure the diabetic anti-diabetic drug is more showing alpha emission than the our synthesized particle, but the result are the positive. These are the graph regarding in vivo study with mice model. The diabetes is induced in the mice with 6 group animals. There are 4 group. This is the streptozotocin drug by which the diabetic is induced, and as a cure we have taken zinc oxide with C1, that is the Tabernae montana, and PC1, that is the Plumeria optimisa, and the sensitivity is found, this is P lesser than 0.051, and this is based on fasting blood sugar model, and there is no drug is given. So animal which are induced diabetic by streptozotocin. Only treatment is given by the particles. The value of significant increase blood glucose level was the 225 milligram per deciliter, and this is the range of the concentration, and this is the comparison with control. Was the difference was 144 milligram per deciliter, and this is with PC1, and this is the glucose level which is reduced by only the zinc oxide which is synthesized with tabernamontanate, that is 344 milligram per deciliter. It means the PC1 is more prominent, more good zinc oxide nanoparticle which can be used as a nanomedicine in anti-diabetic study than the zinc oxide particle which is used as anti-diabetic nanomedicine. That is the amount is this. The amount of increase, that is 500 milligram per deciliter. The compared group is, that is not induced group, was the 144 milligram per deciliter level. Treated with this PC1, this is 213. Treated with only C1, that is 344 milligram per deciliter. And this is the pH at which this study is done, that is 4.5. And the control group is the given only citrate buffer to the animals. This is again one part seed which is studied for antibacterial study of Azeritica indica, but I'm not discussing. This is the third one part, antibacterial study. We did four with at least 18 particle with which we have studied the antibacterial study. I am discussing only with one particle, that is the zinc oxide particle. Shape is rod. Shape is also with L1, L2 was the rod, but with flower extract was totally different. So we can say the shape is oriented by the presence of the phytochemical present in the extract of various part of the plants. Other rest parameter, the rate, RPM, temperature, the precursor amount, and salt, all we have taken the same. That is the Origanum mesoranum, that is Marva plant. Here is we have synthesized the zinc oxide nanoparticle with leaf extract. Again, percentage is 25%. Again, we get the rod shaped particle. These are the leaf parts. Synthesis the same. There are 4 bacteria: Escherichia coli, Streptococcus pneumoniae, Staphylococcus aureus, and Pseudomonas aeruginosa. This is the again result published in Material Science and Engineering in IOP Science. Again, there are some particular phytochemicals. As we know, the antibacterial activity, whatever shown in the synthesized material, that is with presence of the flavonoid. It is the done study. The particle which shows the antibacterial activity, presence of the flavonoids. There are so many other phytochemical are present, but this property is due to presence of flavonoid. Like a anti-diabetic activities, presence of gallic acid. These are the again schematic representation of the synthesis. No need. Again, characterization. The band gap is 2.84 electron volt. The specific things in UV is the particle synthesized with mesoranum, that is, it is absorbed in infrared region also. So this particle can also use as an air emission. The study is in progress with chemistry department, Delhi University, which I am not discussing here. These are the again, I have shown the stability of the particle with help of the XRD. Yes, ma'am. Just I am finishing.
Wind up in next 2-3 minutes.
Yes, 2 minutes. I will take only 2 minutes. These are the again I have shown the morphology. These are the EDS analysis, which shows the percentage ratio approximately 50% oxygen and 50% zinc. That shows the formation of the zinc oxide nanoparticle. These are the antibacterial study we have done with froth dilution method. There are two methods: disk diffusion method, well diffusion method, and other is the froth dilution method. With this diffusion method, we have not get good result, but with froth dilution method, we have find the positive result. These are the bacteria which we have studied against our particle, where our particle act as a antibiotic. And these are the range of MIC in microgram per ml. As it is clear, the MIC value is lesser for gram positive bacteria instead of gram negative bacteria. So we can say, or in our result, the gram negative bacteria MIC value is higher than the gram positive bacteria. And this is depend upon the structure of the membrane or wall of the bacteria, and also the zeta potential of the synthesized nanoparticle. These are the conclusion part. As green synthesized nanoparticle shows very good antibacterial activity against Pseudomonas aeruginosa, Escherichia coli, as well as Staphylococcus aureus and Streptococcus pneumoniae. And the value is higher for Pseudomonas aeruginosa, that is the gram negative bacteria, that is due to negative zeta potential of our synthesized particle. And this is the value for Escherichia coli and Staphylococcus aureus. So our in vitro study with gram positive as well as gram negative bacteria shows that our synthesized zinc oxide nanoparticle with leaf extract of origanum can act as a antibacterial medicine in the future. And there is the recommendation also in a sanitizer, this particle can be used. When I presented some result in MIC conference, then there is the recommendation I can patent this, but still I am not patented. And these are my colleague Smriti Bhatia. This is the student Mona Saini. This is Sapna Yadav. One more my colleague as well as the Nutan Rani. Her work which I have presented here. She is pursuing PhD with me. These are some nano add-on course student contributed something in my some research work. And I also acknowledge to DRDO in Mass, Delhi for anti-diabetic activity, antibacterial activities, CUC Gujarat, Professor Seema Rawat, and for anti-cancer activity, Professor Anju Srivastava from Delhi University Geology Department, and also my student. Thanks.
Thank you, ma'am, for this wonderful talk. We have some questions from Ifra Manzoor. Is this possible due to presence of phytocompounds present in extract that have enhanced or responsible of biological activity?
Yes. Like I have already explained, like anti-cancer activity in leaf part, that is due to presence of quercetin. There are so many phytochemical, but it is the phytochemical. Like flower part shows anti-diabetic presence of gallic acid. There are also flavonoids, but the anti-diabetic property is not because of the flavonoid, but it is presence of the gallic acid. And again, antibacterial, presence of polyphenolic compounds, flavonoid, etc.
Another question from the same. Have you studied the variation in solvent in the formation extract affecting the biological activity?
Yeah, yes. Generally, we don't make any extract in the non-aqueous media because non-aqueous media is not very good for the biological study since our body pH is 7.4. So all extract we have prepared in aqueous media. Yeah, solvent extract percentage, like either we are making 25% in like 50% ethanol and water, or methanol and water, or other solvent. In the similar manner, if we are preparing in aqueous media, the extracted part is different because of the electrostatic attraction with the phytochemical and with the solvent molecule will be the different. So the targeting property also will be different.
Another question, ma'am. Among zinc acetate, zinc nitrate, which one is best for green synthesis for antibacterial, anti-diabetic activity?
Again, please let me know again.
Among zinc acetate, zinc nitrate, which one is best for green synthesis for anti-diabetic activity?
Either we study the anti-diabetic or anti-cancer or antibacterial, the precursor solubility affect the formation of the synthesis, whatever material is there. Either we are preparing nanomaterial or preparing other metal organic framework or other material. Since the solubility of the nitrate is best, so you will get the best result with the nitrate. Though in the formation of the morphology will affect by several physical parameter like time, what is the RPM, what the concentration of the part of the taken phytochemical or extract. But only the metal precursor salt affect the solubility, so yield will be the different. Like zinc sulfate, either taking acetate, taking nitrate has the more solubility than acetate, then least solubility zinc sulfate, but zinc sulfate is very cheap, so we take it. But the activity where we can't say until unless we have taken like acetate, we have tried, we are comparing, so we are taking in the similar manner, we are taking same precursors. But in the UZ experiment, like in DSC course, we have tried this thing, the yield is effect.
Okay, ma'am.
Starting precursor, we take whatever zinc salt is there, either nitrate, acetate, or sulfate, or other. That should be soluble in whatever solvent you are using in your synthesis. That is things, and that should be more soluble. यदि वो ठीक से soluble ही नहीं होगा तो unreacted रहेगा. Unreacted का यदि आप ठीक से washing नहीं करेंगे तो वो आपके powder उसमें भी आएगा और आपका जो भी property है वो clear नहीं आएगा. यदि आपका like if we take the zinc sulfate, if we not wash properly with ethanol, then we get the zinc sulfate as a impurity. So in the powder material, whatever you are using in any type of the activity, the unreacted sulfate show the different type of the way. So whatever salt you take, but washing of your whatever you are getting at the PPT or a colloidal washing should be very good. Unreacted either extract or either the precursor should not be in your final product.
Okay. Ma'am, another question is, dear madam, generally we say green synthesis of ZNO, but for the formation of ZNO, base is required, means NaOH, KOH. But in publication of green synthesis, there is no mention of NaOH, KOH. Then how can we say it green synthesis? And what should be the actual reason of changing band gap energy of ZNO using extract? How does it change?
The band gap is mainly, as we have seen with case of the different extract part on variation, the concentration is effect mainly. Like you have seen in our case with flower extract or with the majorana leaf extract or azuredica, there is not too much difference. But if you are wearing the like in chemical method, then you get the different range. The variation will be higher, but in this case will less. But the phytochemical concentration in our case is affecting. If we are taking higher concentration, then band gap in our case is decreasing.
Okay, ma'am. Thank you
But
for.
in the green synthesis, I don't know, so many researcher or scientists don't show the presence of NaOH or QH. Though there are so many synthesis, QH will not use if we are saying green, but the all green synthesis occur in basic media, or the good way to maintain the basic media NaOH, because it's completely soluble. When we wash completely, will remove unreacted part and cheap. But without basic media, without adding the NaOH, we not get the particles. हमने बहुत सारा कोशिश किया कि NaOH is corrosive, और we are saying like green मे भी कुछ लोग लिखते नहीं होंगे, बट हमने बहुत सारा सिंथेसिस किया है, बट the main thing is pH. In only basic medium it is taking place. तो pH आप चाहे NaOH से करें, चाहे आप अमोनिया सोल्यूशन से करें. हमने ट्राई किया अमोनिया से, बट इट इज नॉट गुड थिंग. अमोनिया के प्रेजेंस में if we are doing only 40 degree centigrade, then also we get the very bad smell in our lab. So NaOH is good कि आपका ऑर्डर तो ठीक रहेगा, लैब का एटमॉस्फेयर तो ठीक रहेगा, और जो भी है चीप है NaOH. और आफ्टर अनरिएक्टेड यदि पार्ट भी रह गया, दैट यू कैन रिमूव बाय ओनली वॉशिंग. Nothing another things you need, no further any reaction.
Thank you, ma'am, for this insightful talk. Over to you, ma'am.
Yep. Thank you, Dr. Sunny. It was really a detailed and a comprehensive talk giving biological activity of zinc oxide nanoparticles. We all know that zinc oxide nanoparticle is a wonder material. It can be used in several non-biological as well as biological applications. So I had a quick question related to where you mentioned about doing some anti-cancer activity using these ZNO nanoparticles using the neem leaf extract. Now we know that there are several compounds associated with neem, like you also mentioned quercetin, azadirachtin, nimbine and all. So have you tried working with the pure compounds and seeing which one
Yeah,
is
ma'am.
showing better anti-cancerous activity?
Yes, pure is like if you get from the market quercetin, then you can try
Right.
only. But if we talk about the green synthesis, the synthesis of the particle, the combined effect of all phytochemical effect. If we separate all one by one, then it is very tedious and very expensive, and very a lot of chemical are used. Though there are methods, we can separate only quercetin from the leaf part. We can separate gallic acid from the leaf part. We have did so many qualitative tests initially in the lab, also some quantitative tests, but these are the test part, or separation process is very tedious, also take a lot of chemical, water as a solvent. So now then it is not environment friendly. So whatever you see in the research papers, the phytochemical whatever part is combined is taken, no one do this work. And the phytochemical whatever present, since the property, like if either anti-cancer property are showing, or anti-diabetic or antibacterial, these are the oriented on the presence of the functional group present in the phytochemical. These phytochemical in our particle is present as a keeping, as a periphery, at a surface property. They are only capping this. So the particular functional group is targeting for particular property. Maybe like our leaf particle connect as an antibacterial also. But one by one step, we did like for zinc oxide or neem. This is the multifunctional. We did the photocatalytic activity, anti-cancer activity, super capacitor activity. Like one paper I am not discussed with Mona Sethi, जो अभी publish हुआ है. So it is the multifunctional material. But ये देगा ये लाइक फ्लावर पार्ट है वो भी क्या एंटीडायबिटिक देगा या एंटीबैक्टीरियल देगा, दैट डिपेंड अपॉन द फाइटोकेमिकल प्रेजेंट इन वॉट एवर अमाउंट दे आर प्रेजेंट एंड हाउ दे आर kept, how they're at the surface of the particle whatever is synthesized, how it is oriented, how it is target to our biological molecule. Either it interact with antibody, either it interact with the like electrostatic interaction like zeta potential of the cell line or the antibacterial wall also. So there are different things, and these functional group will affect all these things like zeta potential is the or hydrodynamic size of the particle in the cell line or in the vivo study is the very important, because the hydrodynamic size, it means whenever solvent in our body fluid or any solvent, then the size of the particle will different than you have synthesized in your lab. So the targeting property as well as its functional property will be different in the biological media as well as in vitro media.
Thank you so much. Is there any question from other panelists? Okay, if not, let us all thank Dr. Kalavati Saini for such a comprehensive talk on biomedical applications using ZNO nanoparticles, green synthesized ZNO nanoparticles. Ma'am, thank you for your talk, and
Thank you,
we
ma'am,
hope to hear more
for
from
having
you
your
in the
committee
future.
also, and I'm sorry for glazing
That is perfectly
on
fine. It
my
happens in all
side.
life. Thank you.
Thank you.
We,
Thank you.
yeah. Over to you, Pooja.
Thank you, ma'am. Next one is one of our young achievers. Our next talk is by Miss Sapna Balwan. She has submitted her PhD thesis and right now working as project assistant under an DST project. She has published in reputed journals. Her research interest in development of biosensor, synthesis of molecularly imprinted polymers, and preparation and characterization of different nanomaterials. Today, her topic will be development of a biosensor for neonatal sepsis detection. Stage is all yours. You have 12 minutes to complete, and after that, 3 minutes of question answer.
Welcome, Sapna. Pooja, is Sapna there?
1 minute. Yes, Sapna is here.
I think IT person, can you please make her the co-host? Yes, thank you. Sapna, please start.
Good evening to all. Thank you, Pooja. Myself, Sapna Balan. I'm pursuing my
Sapna, your
PhD.
voice is echoing. You have to manage the sound.
Okay, ma'am. Good evening to all. Myself, Sapna Balan. I'm pursuing my PhD at American Institute of Nanotechnology. The topic for today's presentation is development of a biosensor for neonatal sepsis detection. Neonatal sepsis is a global concern.
Sapna, can you please turn on your video as well?
Neonatal sepsis is a global concern. Neonatal sepsis is described as a blood infection that is occurring in the new infants during the 90 days of life. Based on the age factor, neonatal sepsis can be characterized into two categories, that is early onset neonatal sepsis and late onset neonatal sepsis. Early onset neonatal sepsis deals within the with infection occurring within the 72 hours after birth, whereas late onset neonatal sepsis is when the infection is occurring within the 90 days of life. The cause of neonatal sepsis is transmission of microorganisms from the environment as well as during the delivery. The early onset neonatal sepsis is occurred due to the various factors such as maternal fever and the transmission of microorganisms during the delivery process. The late onset neonatal sepsis is occurred due to the factors from unhygienic condition from the environment and very low birth weight. There are various bacteria that are responsible for neonatal sepsis, such as E. coli, group B streptococcus, Staphylococcus aureus, Listeria monocytogenes. Neonatal sepsis is a non-specific condition. During this condition, there are various signs and symptoms that are observed. Some of them are fever, lethargy, irritability, seizure, cold, and many others.
Sapna, your slides are not in slideshow mode.
From my side, it's in slideshow mode.
Please carry on then.
There are various biomarkers that have been reported for the diagnosis of neonatal sepsis. Some of them are serum amyloid, C-reactive protein.
Sorry to interrupt you, Sapna. Have you changed your slide?
Yes, ma'am.
I don't think we are able to see a change in the slide. Can you please reshare? Yes, now it has changed to the introduction slide.
Ma'am, now is it on third slide?
You have to speak louder also. We are not able to hear you properly.
Ma'am, can you see the third slide now?
Yes, Sapna. Please carry on.
Neonatal sepsis can be detected through clinical diagnosis as well as laboratory-based methods. The traditionally available methods for neonatal sepsis have various limitations, such as they are time-consuming, expensive, and large sample volume is required for the diagnosis purpose. The sample preparation for in traditional method is very complex. Also, they are not portable. These limitations can be overcome with the introduction to biosensors. Biosensors have various advantages over traditional methods. They provide rapid results. Low cost development is required for biosensor development for the detection purpose. The sample preparation steps involved for the sample preparation are very simple. The device is
Sapna, please reshare your slides, I would say.
Ma'am, can you see my slides now?
Your voice is too low, Sapna.
Ma'am, I have reshaped the slides. Can you see it now? Is visible? Ma'am, can you see the slides now? I have reshared the slides.
Sapna, am I audible? Can you hear me, Sapna? Sapna, am I audible to you? Sapna, you are muted.
Ma'am, is I'm audible now?
Yes, please.
Ma'am, my
Carry.
slides are visible. Okay.
You're on the fourth slide.
Ma'am, I have exited to fifth slide now. Ma'am, my slides are visible now. Fifth slide. I'm on fifth slide.
Sapna, please carry on.
To overcome these limitations, the introduction to biosensor can facilitate the biosensor helps in instant result. The the development cost for the biosensor is very low and very less sample volume are required for the diagnosis purpose. The steps involved for sample preparation are very simple. Also, the biosensor specificity and sensitivity and are user friendly. In the present study, we have developed an electrochemical biosensor for detection of C-reactive protein, which is a biomarker for neonatal sepsis. Step 1 shows the modification of electrode with gold platinum nanoparticles. The gold platinum nanoparticle enhance the conductivity and catalytic activity of the biosensor. In the step 2, the electrode was modified with molecular imprinted polymer synthesized for C-reactive protein. Step 3 shows the washing of molecules from the surface of the electrode to obtain the imprints of C-reactive protein. And in step 4, the electrochemical measurements were performed. The surface of the electrode was studied with scanning electron microscopy. Figure A shows the modification of electrode with gold platinum nanoparticles. Figure B shows the deposition of molecular imprinted polymer on the surface of a modified electrode. The similar type of structures confirm the presence of molecular imprinted polymer on the surface. Figure C shows the imprints of C-reactive protein on the surface of the electrode. The activity of the biosensor was studied with electrochemical measurements. First of all, we carried out the cyclic voltammetry for different modified stages of the electrode. First bare condition was taken where the oxidation and reduction peaks were lowest. Further, with modification to modification with gold platinum nanoparticle, the oxidation and reduction peak enhanced. When the electrode was coated with molecular imprinted polymer for C-reactive protein, the oxidation and reduction peaks decreased due to the presence of polymer on the surface of electrode that resulted into higher resistance and low conductivity. In the fourth step, we washed the template from the surface of electrode to obtain the oxidation and reduction peaks increased again because the imprints created on the surface of the electrode leads to the high diffusion rate and the higher current is generated. The electrode was evaluated with varying concentrations of C-reactive protein within a range from 0.1 nanomolar to 500 nanomolar. Figure B shows that the differential first voltammetry study for varying concentration of C-reactive protein, and it was observed that at higher concentration, the peak current value decreased due to the large number of molecules present on the surface of electrode, leading to the higher resistance. The stability of the biosensor was determined with the scan rate studies within a range from 20 millivolt to 140 millivolt per second, and it was observed that with the increasing scan rate, the oxidation and reduction peaks increases simultaneously. This shows that the system is stable. The activity loss of the biosensor was studied with different interfering compounds, and the result shows that the activity was below 15%. So we can say that the biosensor is highly specific to the C-reactive protein. We evaluated the electrode with different parameters. For pH study, a range of 5.5 to 8.5 pH was obtained, and the highest current was observed at 6.5 pH, which therefore we consider the 6.5 pH as the optimized pH for the developed biosensor. As beyond 6.5 pH, the hydrogen bonds were created on the surface of the electrode, which leads to the higher resistance value and decrease in the current value. The electrode was evaluated within a temperature range from 10 degrees Celsius to 70 degrees Celsius, and higher current value was obtained at 30 degrees Celsius. Therefore, the 30 degrees Celsius was observed at the optimized temperature for the developed biosensor. To test the repeatability of the biosensor, we prepared 5 similar electrodes at similar conditions, and it was observed that the current difference was between 5 to 10 microamperes. So we can say that the system shows good repeatability. Conclusion: the surface of the printed electrode was coated with nanomaterials to enhance the property of the biosensor, such as electrical conductivity, catalytic activity, and sensitivity. The sensitivity of the biosensor for C-reactive protein was calculated at 0.14 microampere per nanomolar. The response time for developed biosensor was observed less than 5 minutes, and the sample volume required was only 50 microliter, which is an important concern in case of neonates. Future prospects of the present study are the biosensor can be used as home-based monitoring. Newer and more regular approach can be developed to improve the early detection and treatment effectiveness of neonatal. Thank you.
Thank you, Swapna. Now we can take some questions.
I think, Pooja, we are running short of time. So thanks a lot, Sapna. Those who wants to interact with Sapna, Sapna, you can please send your email ID in the chat box. You can ask the queries to Sapna through email. I hope that is okay with you because we are really running short of time today. Okay. So Pooja, can we move on to the next presentation, the last presentation of the day?
Yes,
and before we start the last presentation, I would like to announce that there is an assessment, online assessment via MS Forms, so which I'm going to float it very soon. And that link will be active up to 7 p.m. But we will be closing the session at 5:30 sharp. So I will insist that everybody please copy that link, and you can take your time to submit the assessment. Whatever we have learned throughout the day, the questions will be based on that. Okay. So over to you, Pooja.
Yes, ma'am. Thank you. This is the last talk of this day. That will be by Miss Sringika Soni. She is pursuing her PhD from Amity Institute of Nanotechnology. She is working on electrochemical biosensors for narcotic detection. Her research area is analytical chemistry for nanomaterials and biomaterials with potential applications in diagnostic and therapeutics. In her tenure, she has published several research articles in reputed journals. Today, her topic will be aptamer-based sensors for disease detection. Over to you, Shringika. You can conclude your talk in 12 minutes, and after that, some question answers.
Welcome, Shringika.
Shringika.
Can you hear us, Shringika?
Hello.
Shringika, there is an echo in your voice. I think you have to move out of the space where you are sitting right now.
Ma'am, can you hear me?
Yes.
Okay, ma'am. I'll share my PPT.
And please be louder.
Okay, ma'am. So, can you see my slide?
Yes, Shringika. Please put it onto slideshow mode.
Yes, ma'am. It's in slideshow mode. Okay. Good evening, everyone. My name is Shrangika Soni. I am pursuing my PhD under guidance of Dr. Nidhi Chauhan from MIT Institute of Nanotechnology. My topic of my PhD is the aptamer-based sensor for different biomarker detection, and currently I will be talking about the aptamer-based sensor for disease detection or any other drug or other antigen detection. First question is, what is aptamer? My slide is moving, ma'am? No?
Pooja, can you see the slide moving? I am not able to.
No, ma'am, the slide is stuck.
1 minute. First question is, what is aptamer? Aptamer is basically a part of a DNA is sequence of oligonucleotide, which is very fixed, very selective for particular target. That could be anything like antigen or drugs or any disease biomarker, protein or anything. Major advantage of using aptamer from over DNA or immunosensor is aptamer is highly selective and it can bind with any material or small molecule, whereas DNA work on the principle of hybridization, whereas aptamer bind with the weak interaction bond to the target. It is highly stable in comparison to the immunosensor, and recently it has been aptamer has been used in different aspect, like in environmental to detect metals and drugs, and so there are many aptamer based sensor for cocaine detection, whereas other if we talk about the other narcotics, they are very limited aptamer based sensor. So aptamers are like made up of apters, that means 2 fit, and mer is the smallest unit of repeating structure. It's range between 2 to 80 base pair. It is a single stranded folded oligonucleotide and peptide that can bind to the target in a specific manner. It is highly selective and with high affinity with low immunogenic property. It is very small in size and it can be easily modified chemically. Aptamer synthesis is generally performed by the Salix. Salix is a systematic evolution of ligand by exponential enrichment. In this process, I can show you this
Salix
video.
is a process that determines aptamers that are most suitable to fit with the target molecule. There are 4 steps in each round of the Salix process. The first is binding, where some of the DNA strands physically bind to the target. Then you remove the target molecule. That is called elution. The next step is PCR. Here you make copies of the good binding DNA strands, but these copies are now in double helix DNA strands. So we need to get rid of the complementary strand in a process called digestion. That was one round of the Salix process. Now we have a lot more single-stranded DNA that nearly match the target. We have a better chance of finding the DNA strand that will be our aptamer. The Salix process is repeated over and over again until we find a single-stranded DNA which best binds the target.
As we can see, as we saw in this video, that aptamer synthesis is a repetitive process in which we have to, initially it was bound with the target, then bounded oligonucleotide eluted out, then it was amplified, then that amplified product was again incubated with the target, and the same process repeated over and over at least 8 to 10 times, and at last a range of 5 to 10 nucleotide aptamer sequence was found, and that could be further cloned or sequenced, and in which 1 or 2 aptamer sequences can be used for further processing, like in with different application. Aptamer target binding process in this figure, you can see the aptamer target binding process where the RNA or single stranded DNA is folded in particular manner in 3D structure, and it directly bind to the target with high structural compatibility and with weak interaction bond. If we talk about the aptamer-based DNA sensor, it can be divided into 3 parts, where first is direct detection, second is split indirect apta sensing, and third is exonucleases-based apta sensing. In direct detection, aptamer was directly bound to the electrode, and the target was allowed to incubate with the fabricated device, and we check or record the electrochemical signals. In split apta sensing, it is like indirect ELISA, where aptamers are divided into 2 parts, and the main recognition part of the aptamer is half in one probe and another half is another probe. So it was allowed to incubate with the target, and both these split aptamer form a large 3D or 4D structure, and it can be the signal, electrochemical signal, were further recorded. If we talk about the exonucleases based apta sensing, in which exonuclease enzyme play the role for aptamer division and the direct binding to the target. The major difference between aptamer and antibodies are aptamers are highly affinity. It has high affinity and highly specific for the target, and it is in vitro chemical process, whereas antibodies in vivo biological protocol, which has very large further purification and isolation from the host cells and different protocols and experimentation. Whereas in aptamers, binding parameter could be modified, like incubation time, their structural modification, aptamers modification or functionalization with the nanomaterials or any dye or any enzyme. And it has large storage time with low immunogenicity, whereas antibodies are it has very low storage time. After sensing application in the medical diagnosis there are very limited studies has been reported in after sensing like neonatal sepsis, bacterial infection, protein deficiency, tumor, neural diseases, and recently aptamers has been identified for the S and N protein of the SARS-CoV-19. We have published these articles in from our lab where we show the we developed an aptamer-based sensor for amphetamine drug detection. And there is another review in Journal of Electroanalytical Chemistry, which shows recently developed apta sensor for different illicit drug detection like cocaine or opioids and methamphetamine and other narcotics. Thank you.
Thank you, Shringika. Pooja?
Yes, ma'am. Now we can take some question or we can
Is there any question from the audience? So Shringika, that was a very nice presentation. I know aptamers are well researched and can be very versatile. I think probe for any type of disease detection. And in fact, the disease you are working on right now is quite important. So well done. Congratulations.
Thank you, ma'am.
Any question? Any question from the panelist? Okay, if not, I must congratulate all.
ma'am, there is no questions.
Okay. If not, then I must congratulate all our young achievers of this session who have been working very hard to reach their goals. Some are postgraduate students, some are PhD scholars working day and night towards their PhD thesis. I wish you all the best for your future venture. Okay, so this brings us almost to the end of this post-lunch technical session. Now, before we wind up with today's session, I would like to invite Dr. Jagjeevan Mittal to summarize the free lunch session. Dr. Mittal?
Yes, yes. Thank you, Dr. Raman. In the pre-lunch session, there was three.
Your voice is quite feeble.
Now it is okay?
Less, but okay, you carry on.
Okay, so in the pre-lunch session, there was three eminent professors give their talk about different aspects of nanotechnology. First talk was Professor Shankar Ram. He talked about the smart nanostructures, nanotechnology, and their social impacts. So he explained about nanostructures, important properties required for their stability, biocompatibility, functionality, synthesis, particularly green synthesis. He stressed that this can be very good for the social, our society, and then with the problems regarding today we are facing. So he explained an application of nanomaterials or nanoparticles in the supercapacitors, day-to-day life, etc. Another presentation was Professor Rajiv Prakash. He talked about smart the technology for societal application. Sorry for that. So he explained about nanomaterials, of confinement in nanomaterials, and different types, different direction is 0, 1, 2, 3 types of quantum nanomaterials. They are regarding from nano, quantum dots, and then also nanotubes, etc. For this, and then the development of electronic property or the function of electronic size, he explained very nicely. He also explained the top-down and bottom-up methods for developing nanoparticles. He also describes about the micro, nano, platforms and devices based on nanomaterials. And their third was presented by Professor P.C. Pandey. He described about luminescence study of some doped LED, WLED applications, doped nanomaterials. He explained about the phosphorus. This is strontium molybdenum oxide and zinc molybdenum oxide, phosphate oxide. So he explained about red phosphorus. So also he explained then how uh, the lanthanum element doping in the organic phosphorus, it will increase their properties. So he about the strontium, it's different ions like bismuth or like boron and others in the nanomaterial, in the inorganic materials, for how their changes in the properties. Thank you very much.
Thank you, Dr. Mittal. So let me quickly wind up today's post-lunch session. So for today's post-lunch session, we began with Dr. Rohit Kaddam from ISER Bhopal. He talked about lithium-ion batteries and sodium-ion batteries, and he also mentioned about some hard and soft carbon nanomaterial-based anodes. And this topic was really a very much practical topic towards sustainable goals. This is a theme of our workshop as well. The next lineup was Dr. Manika Khanuja, where again she talked about a very important topic about cleaning water or especially dye degradation using photocatalysis technology. And she had been working on MOS2, WS2 and graphitic carbon nitride based nanocomposites. The nanocomposites they've been making with polyaniline, and she had shown some excellent results for her work. The next talk was by Dr. Kalavati Saini from Miranda House, Delhi University. She showed her work, a very elaborative work on synthesis of green synthesis of ZNO nanoparticles and how these have been used for anti-cancerous, anti-diabetic, and antimicrobial activities. And then we had the Young Achievers session from our institute, which had 3 talks. Like it started with Mr. Abhyavatin Selvam, and he talked about the photothermal desalination technology. Again, a very important topic. And the next topic was on biosensor for neonatal sepsis detection. We all know that sepsis is a disease which can occur anytime, and especially when it comes to neonates, it's very difficult to handle. So Sapna has been working on this fabricating biosensors for neonatal sepsis. She has done a very good work. And followed by the last talk by Ms. Shringika. She showed about the very versatile probes in biosensor, that is on aptamer. She showed very nicely how these can be synthesized and characterized. So I again congratulate all the young achievers of our institution, and I hope that all the audiences really enjoyed today's entire session. We would like to really hear from you. We are still online for another 10 minutes or so. We would like to hear from you through the chat box. You can send messages, whatever you want. I've been trying to answer most of your questions, and the link for today's assessment has already been shared. You can try out the questions and see how much you could understand. Even if you did not understand, I would still like you to attempt. That will give us an idea how much you have understood and what more we can do for you to give better insight about nanotechnology. So while we are still online, can we, Professor Sinha, are you online?
Yes, Rana.
Yes. Can we hear from you? Some closing remarks?
Yeah, yes. Thank you very much to all the participants. So making this day a great success. From the morning, we have 100+ participants, and this is really remarkable. And I am really thankful to all the speakers who have taken the pain to delivered the talk even with their busy schedule. And basically, the motto of this workshop is to give the value addition to the researcher who is basically working in the nanoscience and nanotechnology. And this is not just like a lecture or getting a certificate. This is basically a effort to give you the knowledge that how you can use the nanotechnology for your own research or for your study, where you can utilize the advantage of the nanotechnology, and you can get the better response, or you can get the better solution of the existing problem. So this is the basically the whole motto behind our such kind of the workshops or the webinars. And I hope the people who are present there will be continue with the same enthusiasm, and they will be get this kind of stuff for next 3 more days. And we promise all of the participants that if really we find some good speakers or the good participants who have interested to join our team as a speaker, as a voluntary giving the lecture, subject to meeting our standard criteria. So next webinar or the next activity when we are planning, you can drop a mail to us, and we will certainly, our team will be certainly look in that one. And if found suitable, we'll try to give you a such kind of a slot so that we can also expose yourself to the this nanoscience community. As you can see, in this time we have given the opportunity to the young researchers who have really done something or some better work compared to their other colleagues, and that's why we have keep around 1.5 hours for the this young achiever session. So with this, thank you very much, and I hope you will be enjoying the next 3 days more.
Thank you, Professor Sinha. In fact, yes, this was a really great idea to offer opportunity to our young achievers to speak in such a big platform. As I said that, as Professor Sinha also mentioned, that if anybody is willing to collaborate, interact in any manner, or even you want to be a speaker in our future events, so please feel free to write to us. To our website is www.amity.edu/AINT. You can write to our director. I'll just type in the director's email ID. And so as I said, I will remain active for another 5 minutes or so. So Dr. Mittal, would you like to say something?
Yes, sir. Link for today's assessment is provided in the chat box. Kindly fill the form for assessment, what has been done today. So about the presentation, how you can recall all the aspects of the nanotechnology which the presenter presents. So please fill the form. And then, yes.
Ranu, I will suggest the link you can keep active until midnight. Okay. So
Okay,
maybe there are the people
sure.
that at the end of the day they need to fill this assessment.
Okay,
After that, by default the date will change, the assessment form will be over. Okay. So from next time, if you can extend the time, that's fine. Otherwise, from the next time we go up to.
I can do that now also. I have I have extended the time by another hour.
You just give till 11:59 p.m. so that the moment it will be 00 hours, it will by default it will close.
So I've kept it active up to 11:15 p.m.
No. Okay. Thank you very much, all. See you tomorrow at the same time.
Okay, so tomorrow's event again begins at 9:30 a.m. Please be present through the same link. Thank you, everyone.